Hi everyone, thanks for coming. I'm Joby from SBT LabTech. Just going to give quick introductions and then hand over to our speakers for this workshop. We're going to learn about the uses of Dragonfly in a couple of different drug discovery settings. First up is going to be Tina Minnick from Merck. Worked in the pharmaceutical industry since 2010 in multiple companies and CROs over the years, often working with a range of liquid handling instruments, so she has a lot of experience. Working in virus, cell pharmacology, biochem assays and compound management. And over the last 4 years she's been in an automation group at Merck, working on the target validation stage and lead off. Then we hand over to another lady from Merck, Liudmila, senior scientist in the quantitative bioscience department, also at West Point, working on assay development, mechanism of action studies.
Hi everyone. So my name's Tina Minnick. I work at Merck at West Point, Pennsylvania. Right now I'm liking San Diego a lot more than West Point, Pennsylvania. But I work in a medium throughput screening lab, so it's an automation lab. I do a lot of compound management and we run a lot of assays in our lab. So we have a few large assay robots. One of our newest instruments is the Dragonfly, so I was going to go over how we use that. Just to go over the Dragonfly really quickly, it has 10 pipetting channels, so you would just snap on disposable pipette tips when you're ready to go. And it's really nice because the cleanup's very easy. Everything's just put the tips on, throw the tips away at the end of your run. There's a reservoir tray, so the reservoirs are also disposable. A nest for the assay plate, and that nest will move underneath the pipetting channels when you're ready to go. There is an incubation lid if you have a need for that as well. So we use this for assay development. Liudmila is going to go over that after me. I mostly use the Dragonfly for low throughput assays. It's really good if you only have one plate or just a couple plates. I like it because, again, it's a pretty clean, accurate dispense, and it's really easy cleanup. You don't have to wash anything, you know. So an experiment I did on the Dragonfly was I have a 2-day biochemical assay that I run, and we normally use the bioreactor to run this assay. So we use 3D4L plates, and we have the bioreactor dispense enzyme into the entire plate. So it will add 4 microliters per well across the entire plate, and at that point we'll take the plate, put it in a room temperature incubator for 30 minutes, and then we'll add substrate. It's a 4 microliter dispense across the entire plate, and after that we'll put the plate into a room temperature incubator again for 20 hours. It's going to sit overnight, and then the next morning we'll come in, we'll add 8 microliters of quench to the entire plate, a 75-minute quick room temperature incubation, and then we'll read the plate on an Envision. So what I did was I programmed the Dragonfly to run this entire assay without, just unattended, so it wouldn't need me to take the plate, take it off, put it in the incubator. So what I did was the Dragonfly will add my enzyme to the plate. It will put my plate under the incubation lid. So you see the incubation lid in the back. It has a lip around it, so it can kind of sit under there for 30 minutes. So each of these steps have timing maps. So you could tell I want the plate to sit under this incubation lid for 30 minutes. Then at the end of the 30 minutes, it's going to add your substrate, and it will put the plate back in the incubation lid for 20 hours, and then the next day add your quench. After that point, I did have to read my plate on the Envision, so it was really nice having the Dragonfly do this for 2 full days without needing me to do anything. I just set everything up in the beginning, hit play, and then I could let it go. So this is the exciting slide for me. So it's how the data looked, Dragonfly compared to the bioreactor. So if you look in the first column, it's the EC50 values we got for each compound run on the Dragonfly compared to the EC50 values run on the bioreactor. So it says solar right now just because that's what we named our instrument, but you could see how close they are, and on the right there's a correlation plot of how the comparison turned out. So we got 0.998 value, and a value of 1 would have been perfect. So this was awesome. This was really exciting, just knowing it could do this and run for 2 days unattended. So pros and cons for each instrument. The Dragonfly has a very low dead volume. I think it's about 200 microliters. It's really easy to set up, easy to clean up. You don't have to worry about doing any wash. It's really user-friendly software. It's great for assay development and for low or medium throughput assays. Also, no utilities required for this. The Bioraptor pros is they're capable of larger reagent volumes. So we use bottles that can hold 60 mils, so that's a lot larger than the Dragonfly. The reservoirs on the Dragonfly can only hold 10 mils, although there is auto-feed bottles now, so in the future we will be able to do the larger volumes on the Dragonfly. Cons, the Dragonfly is a slower dispense time. You can split your reagent up if you want to use multiple tips to make it a little quicker, but overall it's a slower dispense time than the Dragonfly, and the reservoir limitations is another. So the Bioraptor requires utilities. You have to have an air supply to run them. They're at the end of life, and the software I think is pretty glitchy. Also, the dead volume is pretty large, about 3 to 5 mils, which is a lot greater than the Dragonfly. So path forward, we're talking about putting the Dragonfly on one of our assay robots. This is a picture of our old assay robot. So we have a giant track down the center, an orbiter arm that can pick the plate up and move it from instrument to instrument. So you can see on the left, there are 2 Bioraptors. Behind the Bioraptors, there's a multi-drop combi. In the back corner here, there's a Bravo for plate-to-plate transfers. We have 2 plate readers. There's an Envision and a Ferristar. Incubators underneath. So there's a room temperature and a 37-degree incubator underneath the system, and plate washer for cell-based assays. So maybe in the future we can add the Dragonfly to this. And my last slide is an art and science challenge that I did in September. There was a Merck Technology Symposium. So I had a lot of fun with this. This is 6 3D 4-well plates just super glued together, and I had to make different plate maps for each of these. Each color could represent a different reagent. I actually learned a lot about the Dragonfly using this, because you could use CSV files for complicated plate maps, so you don't have to have one addition to the entire plate. So for custom plate maps, this would be great. And even the reagent volumes can be different from well to well. They don't have to be the same volume. So this was a lot of fun. I was hoping it would be like more of a competition, but it ended up not being one. I thought I was going to win a prize. I think I would have won with this. And that's my last slide. Are there any questions for me?
Hi, everybody. Thank you. Thanks a lot for inviting me to talk about our applications for Dragonfly. So Tina just explained nicely about the instrument and what the utility is. So I'm going to go and just give more examples how we use the Dragonfly for assay development specifically. So actually, I was really excited when the first time I saw the instrument, because as soon as I saw the special software, Dragonfly Designer, and I saw what it can do for assay development, it was like the right next day. Probably I jumped on and tried it, because I do a lot of assay development, and those of you who are familiar with this know that it's a lot of manual, labor-intensive work, and it takes several days to complete the task for the validation and characterization of specific format. So this way, by using the Dragonfly, it was so very convenient, because it's very flexible with the plate map development. So you can repopulate your ingredients, substrates, buffers, enzymes, and since it has different channels, like for example, we have 8 channels, you can actually repopulate all of the channels. So it gives you excellent utility in terms of the diversity for the plate. So for example, here's the example where I divided a plate into the 4 different quarters, and that's also pre-populated in the designer software. So you can do different positioning on the plate, and then you can specify which reagents goes where, and then you do titrations. And it's also very flexible in terms of titrations. You can do your own custom-made titration, or you can do n-fold or linear titration. and you can do titration from vertical or horizontal. So that way I was able to multiplex my assay on one plate, and I was able to get basically KM for 2 different substrates and also linear enzymatic titration. On one plate, like within a couple of hours, I get the data, analyzed it, and that saved me lots of labor and time. So that was very exciting. Another thing which I want to add is that because you have multi-channel, so you can speed up the process of addition. And if, for example, in my case, I'm doing continuous assay, it actually helps with the timing so that I can measure my time correctly. So next problem we had is also that actually Dragonfly was able to fix for it is because of the low volume, you can actually pre-test different proteins. For example, if we select a protein for your assay and different vendors make the proteins, you can actually test them on one plate. And for example, we were able to test protein activity of different vendors on one plate and select which one is actually more suitable for our applications. So that was actually another great application for us. So right now, and the good thing about this is that all of the Dragonfly designer protocols, you can save them, and then it's easy to use in the future, so that initially it may take some time to pre-populate all of this information, how you do titrations, what the volume is, but then if you're going to use it later on, it's very quick. You just upload it and you're good to go. And it gives you all the information you need. For example, if you want to only, if you know all the concentrations, so it can pre-populate what the volumes you need to use. And also it tells you actual what the concentrations you need to prepare your stock solutions for. Also, you don't have to care about buffer or bulk volume because it pre-populates that too. So that saves lots of time with math. So, and then in my last example is just we do a lot of MOA because of this, because we can titrate it, compounds or substrates or enzyme. So for example, we were able to do Dixon plots and to identify competition assay for substrate competition assay in our application. So overall, I would say that it works really nicely for assay development because it has low dead volume. So that means in the initial stages of the assay development, when you don't have lots of reagent, or you're just testing a system, a format, or detection system, then it actually saves a lot of money for that. And the same goes for the protein QC or validation, that it's because of low volume, it helps with that. And then also because you can actually, when you're on the assay development stages, because of the limitation of the materials and reagents you have, you don't have to use whole plates. You can only select a small region of the plate and work with that. So that overall gives us lots of advantages and saves time. Yeah. So thank you.